going to start with Nacho. He's going to give us a speech regarding monoclonal antibodies, the development of monoclonal antibodies, and maybe the future of these new drugs, this new modern immunotherapy. At the end of the two speeches, we're going to discuss about their thoughts, and there are some new concepts in immunotherapy. Thank you, Nacho.
Thank you very much indeed, Luis. When I got invited to this, there were a completely different panel of speakers, and actually what I'm going to do, hopefully I'm going to do, is to get you through what has been a revolution in immunotherapy, taking a little bit of a historical perspective and trying to point out which are the main obstacles that we are finding and what are the challenges we have to deal with on an everyday basis. So the whole field of tumor immunotherapy is depending on some cells that Santiago Ramón y Cajal, our famous Nobel Prize winner, thought were just a result of evolution without too much function. And the reason is that when you look at lymphocytes in most organs at a steady state, it is a boring cell with a lot of nucleus, very little cytoplasm, and nobody knew what they were doing. But obviously, they are the only cells in the body that can acquire the function of killing other cells. There are two cells in the body that accomplish that function, and they are the cytotoxic T lymphocytes and the natural killer cells. And obviously, their function must be very well regulated, because otherwise they would create inflammation and tissue destruction. That's what they are evolved for. The whole idea is that at the price of killing cells, you can limit the replication of a pathogen. And the problem is that when you are facing tumors, which are very wicked cells, what you really need to do is to use a function that evolution has sculpted in the organism for a completely different purpose. A very important thing in our business is that part of these cells with the capability to kill can revert into a memory state and can be called into action again with some degree of efficiency in targeting an antigen that it's specifically recognized by several mechanisms. So in a way, most of the talks that you are going to hear after mine, what they are trying to do is to use these guys, or these guys here, as micro robots that can act in the organism, seeking, recognizing, and destroying tumor cells based on different strategies to confer them with such properties. Unfortunately, in all this scheme, tumor cells have a human genome that they can use. They have a nucleus full of genetic and epigenetic ideas that they can use to tamper with these mechanisms that in this very simple slide are represented by arrows. And all those mechanisms of interference tamper with the immune response in such a way that it becomes ineffective. And a lot of the success of the field of immunotherapy up to date has been just interfering with the yellow arrows. So the story of immunotherapy and cancer has a lot to do with a highly debated idea that was proposed by it was proposed by Macfarlane Burnet and others a long time ago. And the whole idea is that the immune system was endowed with the ability to recognize nascent tumors and eliminate them. And that's called immune surveillance, because they are like sentries patrolling and then trying to find out if there is a tumor, and if there is a tumor, attack it. And that sounds very well, but there was very limited proof. And actually in humans, we are still debating that issue. But in mice, it's much simpler. We know more about the immune system of mice than about the immune system of humans, because we can manipulate their genome by transgenesis, and because we can inbreed them, that's something we cannot do with humans. And in that regard, we know more about the function of the immune system of the mouse than the human, which is Not very convenient, but it is the way it goes. And then if you get outbred strains and you just observe them for the incidence of tumors, as it's expected, very few mice develop tumors. But if you do the experiment in animals which cannot recombine the antigen receptors, the antibodies, the antibody genes, or the T-cell receptors, then there's more incidence of cancer, even in the absence of any mutagens. If you accelerate the process with DNA alkylating agents or any other mutagens, you get more. And also, another thing that seems to be crucial is the interferon gamma STAT1 signaling pathway, because if it is deficient in the mice, then tumors accumulate also without the need of intervention. These are just aging mice. So how to manipulate for immunomodulation the immune system? So we owe a lot to a couple of scientists working then in Cambridge, Cecil Milstein, and George Kohler, who came up with the concept of monoclonal antibodies. Actually, they caused a revolution in biotechnology without meaning to, because what they were looking at is the antibody genes and trying to understand what was going on, and for that purpose, they needed to immortalize B cells producing antibodies. And actually, they did that using a property of some viral proteins that can be mimicked with polyethylene glycol and other osmotic agents, which is the ability to fuse cells. So they fuse membranes. And in that regard, they could immunize rodents, fuse them with myeloma cells that were kind of selected for not producing any antibody. And these hybridoma cells were producing the antibody encoded by the rearranged genes in the mouse B cells. They will divide like the myeloma cell line indefinitely, and you know very well that led to the hybridoma technology, which is the way monoclonal antibodies have been traditionally produced. Now, typically, we use other techniques, but everything comes from this idea of being able to immortalize B cells producing antibodies and clone them to make sure that you had only one antibody, and that's what monoclonal antibodies are. There was no patent for monoclonal antibodies for a number of interesting reasons. Apparently, they intended to, but they eventually did not do it. But there was a very, very clever student in the lab of Cecil Milstein, who was Greg Winter, and he came up with very important patents. He came up with different ways to make antibodies, but using libraries of human, light, and heavy chains for the antibodies, so they would be human variable regions. And the other idea is that he could engineer the Fc portion of the antibodies to make them human. That's what we call humanized antibodies or fully human antibodies. And that was very important therapeutically, because if you inject rodent antibodies into a human being, what happens next is that the human being forms antibody against the rodent molecules, and that interferes with function, is what we call anti-drug antibodies. But with humanized antibodies, this happens less often, and with fully human antibodies is less common, although sometimes, and in some of our favorite immunostimulatory antibodies, can happen as well. And the concept of immunostimulatory monoclonal antibodies, I keep saying that it's very simple, because many scientists had been using antibodies in tissue culture plates to expose immune system cells to antibodies and see what happened after incubating these cells in such a way, and there were many functions that were discovered and studied in such a fashion. But what about if you do it in vivo? And then the concept of immunostimulatory or immunomodulatory monoclonal antibodies came with the idea that we could inhibit inhibitory functions. We can disable inhibitory functions that we know that we're acting on receptors that are antagonists of the activation of killer cells. The other idea is to try to use antibodies as agonist moieties able to trigger activity from stimulatory receptors or co-stimulatory receptors in the immune response. And there are many examples of molecules that can be targeted with these kind of receptors: CTLA4, PD-1, PD-L1, BTLA, LAG3, TGF-beta, IL-10, NKG2A. This list is growing of the inhibitory part and has been by far the most successful to the date. The other idea, and many of the receptors are members of the TNF receptor family, but without the death domain, non-conferring apoptosis once the receptor is forced to signal, typically by cross-linking of the protein, and that belong to the TNF receptor family. You will hear about 4-1BB when the CAR lectures will be given this afternoon, and CD40, OX40, GITR, and CD27, and this list is also growing. But there are approvals in many diseases for part of these moieties, and some of these are also running up. The idea is the control of these killer cells obeys to many receptors. And that's a problem, because if there were only one gas pedal, one brake, and one clutch, we would be able to manage. But there are many, and they are different. So we have to figure out what's the one we need to use. And this is a little bit old from a review of us in Nature Reviews Cancer, but the whole point is that all the ligand receptor pairs listed in this slide that now is growing have samples of antibodies that, if directed to these moieties, are able to elicit a rejection or control the growth of transplanted tumors in immunocompetent mice. So this list keeps growing. And we are in the middle of a revolution. And the reason is that one of the ligand receptor pairs that we will be speaking about, the PD-1 interaction with PD-L1, not so clear about PD-L2, is able to have therapeutic activity, very meaningful therapeutic activity in the neighborhood of the 15 to 30% overall response rate and durable response rate across a huge spectrum of malignant diseases in humans. If you look at this slide where you see the different diseases and the overall response rate that in some cases are dramatic, as a refractory Hodgkin lymphoma, two things hit your brain. One of them is that we are benefiting patients across malignant indications. The other point is that we are underperforming in a series of patients. You can take it in the optimistic way or in the pessimistic way. But this is obviously a revolution that if I would have told Luis 20 years before when I was dreaming about these things, he would have laughed at me, and for a very good reason. But that's the current status of the field with PD-1, PD-L1 antagonist antibodies. This comes at a cost, and the spectrum of adverse reactions mimics some of the autoimmune disease with some dissimilarities to the naturally occurring autoimmune diseases. And the fact is that typically they are self-limited diseases, and the other point is that the reactions are usually very amenable to treatment with very few exceptions. And they come mainly in the skin and in the gut, and with far lower frequency in endocrine glands, in the liver, in the heart, in the bone marrow, etc. And all this spectrum is new to this field, and we need to learn clinically how to manage them, and that's our daily bread and butter. And because we are starting to do combinations and more aggressive forms of treatment, we have to deal with some of these systemic inflammation or autoimmunity-like reactions. So who brought us to the PD-1 and PD-L1 revolution? So it's a very interesting story. There is a book that I forgot to bring with me, which is The Cure from Within, written by a New York Times journalist, and it's about the different people who contributed in the field of immunotherapy. And it's very interesting from the human standpoint and also from the scientific point of view. So Tasuku Onyo, at the time at the University of Kyoto, and that's where he remains, he discovered a very boring family of the immunoglobulin superfamily that happened to be expressed in T cells about undergoing apoptosis. Developed antibodies, developed the deficient mice, played around in the system, nothing really very clear. Maybe the knockouts were rejecting a little bit better tumors, and that's how he posted that. Obviously, that molecule had ligands, and the ligands were discovered simultaneously, almost simultaneously, by Liping Chen, then at Mayo Clinic, and Gordon Freeman in Harvard. And they figured out that that interaction, particularly Lipin-Chen, was able to protect tumors from the immune attack. And the idea was to block that with antibodies and see what happened. And what these people were able to show, Tasuku Onyo and Lipin-Chen, is that that gave rise to a stronger anti-tumor immunity. In a way, all these receptors, importantly including PD-1 and PD-L1, happen to interact in the tumor microenvironment from the T cell with the tumor cells themselves, but also with antigen-presenting cells and tissue macrophages of the tumor-associated macrophages. And whatever happens here can be interfered with antibody, with one limitation, is that antibodies are proteins and do not cross the plasma membrane. And there are many targets in immunotherapy that we know could be very useful that happen to be beyond the plasma membrane, such as transcription factors, signaling events, metabolic events, epigenetics, etc., that we like to be able to selectively act upon. And certainly there are many things that we don't know, and that's the reason probably immunotherapy is still in its infancy. As I mentioned, our main success has been to act on the inhibitory receptors blocking their function. And it's amazing the number of regulatory mechanisms that are inbuilt in the immune system to prevent its overactivation. But there is another way of thinking, which is this idea of enforcing co-stimulation with several biotechnology strategies, that it's also taking place in the form of T-cell engager, bispecific antibodies, antibodies agonist of co-stimulatory molecules, fusion proteins, cell transfected with this and that's very interesting. But the most striking word in the field of immunotherapy is probably synergy, which comes from this mathematical idea of not having an additive effect, but something that is far greater than the sum of the two effects from each of the components of the synergy. And I happened to write something very funny in 2002 in a journal which is published in Poland, but that's because I got stranded in an airport in Poland at that time. So the guy who was the editor of this journal told me, oh, write whatever you want and I will publish, no? So basically I wrote, effective tumor immunotherapy, start the engine, release the brakes, step on the gas pedal and get ready to face autoimmunity. That for year 2002 is great. And the problem is that according to usual rules of pharmaceutical development, you have to use one finger and one hand to control the cockpit of a craft with real capability of destroying tissue. So, you know, it's not that easy to select which is the button that you have to press. But in a more academic fashion, what you need to have is more T-cell recognizing the tumor, probably. That's the best idea. And for that purpose, you can try to use vaccines. This has been very, very successful in many instances. In tumor immunology, we are not really able to get something working extremely well. You can infuse the T-cells after culturing them in the lab, and certainly you can try to turn some of the tumor lesions into a vaccine. It's something we favor a lot in our lab, and for that purpose we inject viruses, we inject locally pathogen molecules, we irradiate the tumor, we provide certain forms to kill tumor in an immunogenic fashion. And that's to start the engine. But once you have the engine going, what you have to do is to tamper with the brakes, and there are a number of them. You can try to act on the gas pedals. No? And then now I live in Pamplona, and in Pamplona, it's northern Spain, you know, we have bumpy and windy and not very good roads, so I believe we have to work in the tumor microenvironment, and that's what I say, that it's acting on the road. No? We need to repair the road. And the overall objective is to get a better tumor response in a synergistic fashion. And there is a cartoon, a plot that I typically updated with the different immunostimulatory monoclonal antibodies, and arrows represented the synergistic effects, whether they are in clinical trials, it has been observed in animal models, or are already clinical standards, such as anti-PD-1 plus anti-CTLA-4 with the nivolumab ipilimumab antibody. But this is very difficult now. And this comes from a review by Daniel Chen and Aria Melman in Nature, where they had an arrow where they plot the different malignant indications which are prey of immunotherapy, and then they look at the different combinations of immunotherapy plus immunotherapy agents, excluding the combinations with a conventional way of treatment, And the first circle is phase 1, 2. The second circle is phase 2B, although we are changing that a lot of the time. This circle here is phase 3, pivotal trials for registration, and this inner core is approval things, which are only in melanoma and now in renal cell carcinoma, and probably soon in non-small cell lung cancer, the combination of CTLA-4 and PD-1 blockade. And this is really amazing, because if you go to clinicaltrial.gov, in the way it is now, there are more than 1,200 clinical trials of IO combinations, and this is truly amazing, and it's kind of a deluge. And in a way, we are probably trying to win the lottery by buying all the tickets, no? Which is kind of interesting in itself. And so here we are, trying to use these cells as micro-robots to destroy cancer. So thank you very much for your attention. And the whole thing is that we are supposed to have interventions from the floor and whatever questions you ask, but I go back there.
All right. So we're going to cover in the next 15 minutes or so the concept of only one of these strategies that was mentioned in the previous talk, which is the bispecific antibodies, and in the context of hematological malignancies. I'm a clinician. I work in Barcelona, and I will be talking mainly from the clinical perspective. I won't go into the details of the immunological mechanisms, although we may cover that in a way in a couple of slides. So these are my disclosures here. So as in oncology, in hematology, we've been shocked by this revolution of immunotherapy in the last 15 years or so. And we've had lots of strategies going on, and we're having lots of strategies going on these hematological malignancies. We started with monoclonal antibodies. The rituximab, anti-CD20, was the first one, like almost 20 years ago. Then we came with more sophisticated molecules or constructs. For instance, the conjugated monoclonal antibodies that are antibodies that are linked to a drug or a toxin that's introduced into the cell and kills the cell. And also we have these bispecific antibodies that are small molecules that they allow to link to different antigens, usually an anti-CD3, which takes the T cell against a wide variety of antigen. In the case of hematological malignancies, being B-cell antigens or myeloma antigens or myeloid antigens for acute myeloid leukemia. We also have bigs and trigs, which are kind of similar of the bispecific antibodies, but are linking not to the T cell, but the NK cell. And also we have the CAR T that will be covered in the next session. But as I said today, I'm going to focus on the bispecific antibodies only in hematological malignancies. There was supposed to be a previous talk on the background, so I won't cover that either. I didn't plan to talk about it. But for those of you who are not familiar with that, the bispecific antibodies are small molecules, small constructs that, as I said, link the tumor cell very close, because they are very small, to the T cell, allowing a direct lysis of tumor cell and also a release of cytokines. And that helps us to understand, as I will tell you later, the beneficial effect in tumor killing, but also the side effects that these treatments may produce to the patient. This is a very nice review that was published a couple of months ago in Blood, reviewing all the bispecific antibodies used in hematology. You have here, as I said, B-cell malignancies, starting for acute lymphoblastic leukemia. The first bispecific antibody approved, both in Europe and the US, was in this disease, but also in other settings, including lymphoma. We also have wide variety of agents in myeloma and growing rapidly, and also in myeloid diseases, mainly acute myeloid leukemia. So today I'm going to focus in B cells and a little bit in myeloid malignancies. I won't cover myeloma. So for those of you who are not familiar with acute lymphoblastic leukemia, this is a disease, is the most frequent leukemia or most frequent cancer in children, and also is present in adults, although from a relative perspective and compared to other malignancies, solid tumors or even hematological malignancies, is less frequent in adults. The prognosis is much different in children and adults. You see here, this is one of my favorite slides in ALL. The prognosis being improved, has been improved over the years. You see here the prognosis in the 1970s that has gone up to over 90% now of long-term survival of these children with B-cell ALL. For adults, unfortunately, the results are not the same you can see here, although we have some good subset of patients, good so-called, with long-term survivals around 60%. We have other populations with very, very good, bad prognosis. And that's for first line in the relapsed refractory setting. Until now, at least, the results are very poor in these patients. All of them require stem cell transplantation, and long-term survival is pretty low. So as I said, in these past 10, 15 years, lots of antibodies have been developed and assessed in this setting. We have trials for CD20, rituximab in first line, which have been associated with better overall survival of patients. We also have clinical trials and drugs approved against CD22, especially inotuzumab ozogamicin, which is a conjugated monoclonal antibodies, and against CD19. And here we have to talk a little bit about blinatumomab, which is the first and actually only one bispecific antibodies approved in hematology now. So the approval came from this trial, which was published more than a year ago now in the New England Journal. And this trial randomized in a 2-to-1 fashion patients with B-cell ALL, adults, to receive blinatumomab or chemotherapy in the relapsed refractory setting. And the endpoint of this trial was overall survival. I won't go into the detail of this paper. I don't think it's the matter of this talk, but just to summarize here, the characteristics of patients are pretty much standard for relapsed refractory ALL, 40 years old, median age. About 40% of the patients had refractory disease. This is a very acute, rapidly progressing disease. And 1/3 of the patients had received allogeneic transplantation before, so very poor population of patients. So, and going directly to the results, what it was seen here is the complete response was more than double in the blinatumomab compared to the chemotherapy. And when we looked at what we call complete remission without complete hematological response, that is no evidence of disease, but we don't achieve, or the patient does not achieve complete platelet or hemoglobin recovery, although it's not dependent on transfusion usually, these rates went up to 44% in the blinatumomab compared to 25% in the chemotherapy group. Also for overall survival, there was a benefit for blinatumomab, which doubled the overall survival, but in terms of absolute months, this benefit was, I would say, moderate. Blinatumomab has been tested also in other settings of B-cell ALL. It has been tried or studied in the MRD positive ALL, that is a presence of a very little amount of disease, but not an overt relapse. But we know that this status is a very good marker of frank progression, and it takes very, very short time from the patients going from the MRD positive status to the frank relapse. And when blinatumomab was given to these patients, 78 of them achieved an MRD negativity, and you see the median overall survival was 3 years. In Ph positive ALL, which is one subset of bad prognosis in the ALL landscape, another phase 2 trial reported results in 35% here CR, median overall survival of 7.1 months. But blinatumomab is also moving into other subsets of ALL in other settings of the disease. And this is, to me, the 6 more important clinical trials going on with blinatumomab right now. You see here the first trial, which is a Spanish trial. We are evaluating the use of this bispecific antibody into the first-line therapy for patients who have achieved a good clearance of minimal residual disease. That's good prognosis, so-called, because these patients have like 35% of relapse risk. So 1/3 of the patients with this good prognosis will eventually relapse. So in order to reduce this relapse risk, we have switched or changed some of the consolidation cycles of conventional chemotherapy with blinatumomab, with this antibody, and we are hoping to reduce the relapse rate. Blinatumomab has is being evaluated as a maintenance therapy after transplantation, also in combination with pembrolizumab. We've heard about these checkpoint inhibitors, and I'm going to talk about them a little bit more in a minute. Also in pediatrics and in Ph-positive ALL. And also this drug has been evaluated in other settings. This is the lymphoma trial published a couple of years ago in the relapse refractory setting, again, of non-Hodgkin DLBCL, diffuse large B-cell lymphoma. 43% of responses were reported in this trial, although it was stopped because of neurological events, which you'll see in a minute, that are a frequent complication of these constructs. As per acute myeloid leukemia, much more preliminary data on that. There are in vitro studies and also mice models that have demonstrated that these bispecific antibodies against CD33, which is a myeloid antigen, very common, are capable of killing cells both with favorite, intermediate, or adverse cytogenetics for acute myeloid leukemia. And also this mouse model you see here, patients or the mice actually receive the injection of the leukemia. Only these ones were treated with the bispecific antibody against CD33. And you see here that all the patients develop leukemia in very short time, while patients that received the BITE stay free of disease. We have to go further, and as it was mentioned in the previous talk, very wide variety of combinations have been tested using immunotherapy and also using bispecific antibodies. One of the most interesting to me is the combination with checkpoint inhibitors, for instance, anti-PD-1, and the combination with BITEs. So by combining these two strategies, these two compounds, we take out the brakes of T cells. So the T cells can act much more deeply, much more strongly against the tumor cells, which allows a high release or destruction of the cells and high release of neoantigens. So these antigens can be recognized by other T cells. And although the molecule is not present in the patient anymore, this can help priming other T cells and renewing the T cell response against the tumor cells. So in a way, there's a way of persisting the BITE activity, although the molecule is not present anymore. But as I said, these drugs have their problems in toxicity. These are the 3 more frequent, or the 2 more and a back of others. Neurological toxicity, very frequent cytokine release syndrome that I'm sure it will be mentioned in the next talk, and also infections and liver complications. Regarding neurological toxicity, this is the most frequent toxicity, up to 52% in some blinatumomab trials, although in others it's around 10%. Clinical presentation is very, very wide, so it can go from seizures to tremors to encephalopathy, and it's usually reversible. As per cytokine release syndrome, we are going to hear about it. I'm not going to go into detail, but what I can tell you is that it's usually presenting in the first cycle, so when there is active disease, and that is less frequent and more manageable than with CAR T. And of course, for liver complications and infections, they have specific treatment and diagnosis. So this is a very busy slide, but it's like a summary of these 7 trials we have with bispecific antibodies in our unit. You see we have a wide variety of trials. You have this trial of anti-CD20 and CD3 in relapsed refractory non-Hodgkin's lymphoma. We have this same compound evaluated with R-CHOP, which is the standard chemotherapy in follicular lymphoma and DLBCL. We have a phase 1 first-in-human trial with anti-CD123 and CD3 in acute myeloid leukemia, and a few more trials in combination of this CD20, CD3 molecule with anti-PD-1 in different settings here and here, and also this first 1 trial in several indications like mantle cell lymphoma and follicular lymphoma, which are less common forms of lymphoma, and also the blinatumomab trial that I mentioned before. So just to conclude, and as an introduction to the next session, these molecules have or are coming at the same time of CAR T in hematology, and sometimes they tend to be compared. So in this slide, I summarized the characteristic of the BiTEs and pointing these differences with the CAR T. So the BiTEs are small molecules as opposed to CAR T, which are T cells with a receptor, as we will hear later. Both are administered IV. For bispecific antibodies, the use is universal. So this is a drug that's in the pharmacy, and you can take it for all patients. But the CAR T are produced at individual basis, at least at the present moment. For the BiTEs, you have immediate availability. You can treat the patient the same day. For the CAR T, you need manufacturing. No lymphodepletion chemotherapy for the bispecific antibodies. You need this chemotherapy for CAR T. However, for bispecific antibodies, we don't have much activity on CNS, at least no data. We know that the CAR T do have activity on CNS. Very short persistent for the bispecific antibodies, very small molecules that last very, very shortly in the blood, while the CAR T can persist during weeks or even more, depending on the construct. And both are approved in some indications, but the main amount of investigation going on or treatments going on are in clinical trials. And all these bispecific and also CAR T treatments in our hospital are taken within this multidisciplinary unit, which is, I think, the way of approaching these patients. You see here all the components of this unit that we call UTA, and have people from neurology, immunology, ID, BMT, ICU, and all types of professionals dedicated or committed to the patients receiving immunotherapy. And if you want to know more of this unit, and if you attend the Spanish hematology meeting, we'll be presenting an oral session. In the oral session, this UTA that we've been doing now for more than 1 year. And I think that's all I wanted to tell you, and thank you very much for your attention.
Questions in the audience, please? Any question? Well, I want to start with Nacho. With respect to the monoclonal antibodies, there has been much more success with the inhibitory monoclonal antibodies, but with agonists, this history has been different up to now. We have some trial with very high toxicity, with no efficacy, or we don't know if they are an efficacy therapy. What is your thoughts about the agonist antibodies?
Yeah, The whole point about agonist antibodies is that now they need to be developed on top of a standard of care, and the standard of care is immunotherapy with antagonists. So there are a number of trials. There is risk of toxicity. We are learning how to co-stimulate in the system in a better way. Biotechnology is advancing. We have now targeted versions of these co-stimulatory molecules, so you get selective accumulation in the tumor microenvironment. We are starting the clinical development of such entities. There are also probodies that get activated only in the tumor microenvironment by means of masking the antibody with a linker sensitive to metalloproteases. So the field is extremely active. The important question is how to optimize their use and how to design trials towards registration of some of these strategies. And certainly pharmaceutical and biotechnological industry is very active with many of these potential functions. But as I mentioned, it's technically simpler and probably to some extent less risky to inhibit the inhibitors rather than exploit agonistically functions of the immune system. But on the other hand, in the mouse models, the most striking synergies, really curative strategies, typically involve agents such as IL-12, 4-1BB, like in the cars, for example, that it should not be out of reach. And that's the whole idea. It's behind combination, but in many mouse models, you really achieve curative efficacy in very tough models. So there's a lot of hope, and there's a lot of there are a lot of clinical trials going on.
Okay, great session. Many thanks. I understand that, well, one of the problems of this immunotherapy is that really the number of patients or the proportion of patients that benefit is, let's say, limited. And considering the cost, selecting the right patients is crucial. And so I wanted you to elaborate a little bit about the future of biomarkers and ways, methods to select the most promising patients. How is that developing, and how do you see the future about that?
Yeah, probably we have to ask both to answer to that question. So biomarkers are critical. And we need them in order to select patients for clinical trials in such a way that we would address the treatment to the right population. We need them to save side effects that won't be needed. We need them to detect the patients early who are going to progress in order to take them to the next option of treatment, including, importantly, clinical trials, and as you mentioned, to save financial toxicity as well. So we are having a number of parameters, multiple parameters that we can use to enrich the fraction of patients who respond, that include several things, but mainly the status of the expression of the targets in the tumor tissue, the level of T-cell infiltration in the tissue, the gene signature, the gene expression signature of genes involved in T-cell activation pre-treatment, the tumor mutation burden in transcribed exonic sequences, because these are the raw matter from which antigenic epitopes are made, neoantigens. And also we are learning things such as the involvement of the microbiota in telling you who is. So some of these are simpler than others, but we are trying to use them probably in a compiled fashion in order to identify patients. That's going to advance dramatically in the next years, and it's obviously a very important chapter. I don't know if you would like to add something on that.
Well, I fully agree with Nacho with respect to these biomarkers. There are a wide set of biomarkers that we have to analyze in the future. Up to now, there are very few that we can rely on. I think maybe PD-L1 in some point, and the first line in non-small cell lung cancer, maybe TILs in breast cancer, but in rich populations. So it's difficult. We have to analyze a wide set of biomarkers in the clinical trials that are running now. And in the next future, we have to introduce these therapies targeted therapies in targeted populations, because this has to be in this way. Otherwise, it is going to be a disaster, not just in the clinical setting, but economically it's going to be impossible to sustain.
Just to add one comment, is that to develop a biomarker to be clinically meaningful needs a process that it's also expensive and cumbersome, which means the generation of what we call now a companion diagnostic or a complementary diagnostic. And it's complex because you have to make actually sure and validate prospectively in series of patients that it works, and becomes almost as difficult as developing the drug itself. And this is giving a hard time to the translational medicine departments in big pharma. And certainly the biomarkers the community of oncology is used to are yes and no, like you are pregnant, you are not pregnant, and you cannot be in the middle. But most of the biomarkers that we are identifying in the immunotherapy space are continuous variables where you have to set up, you have to predefine or define cutoffs and use them as a quantitative variable. And that's giving us a very hard time.
If I can add a comment, for bispecific antibodies, and also this might be true for CAR-T, beyond these biomarkers, we need to know what type of T cells are linking to the tumor, because we have data from a lab that there's a wide variety of T cells going on into the patient with the same malignancy. So we are engaging this T cell, which might be a regulatory T cell or a predominant effector T cell. So the response might not be the same in the two. And that's also true for the CAR-T, that we are sending some lymphocytes to expand and transduce, but we certainly need more efforts into describing these populations of T cells in order to know what is doing the job exactly.
Perfect. There's a question there. This is the last question because we don't have more time.
Yeah, because T-cell immune response is quite heterogeneous. Are we doing like single-cell RNA-seq to really understand trajectories and heterogeneity within the T-cell populations? I think there is a Nature paper in breast cancer. I don't know. Do you think this is relevant? Because I think studying T-cells as a bulk, I don't know if it makes sense in understanding, immune responses in different individuals.
I'm aware of a number of initiatives to get as much information from the tissue as possible in order to figure out, because if you are doing really single-cell RNA-seq, one of the things is that you are interrogate the system in a blinded fashion, so you can end up with signatures that you would not have anticipated and happen to be very important. And that's, for example, something that Tony Rivas pioneered in melanoma. As you mentioned, there is a recent paper in breast cancer, and there is also I guess that is still a manuscript, but there is very interesting information on glioblastoma.
Have to move forward because we are 5 minutes past 5, so we have to move forward to the next session, the CAR-T cells. So the speakers, thank you so much for your attention.